Cancer. What is cancer? A definition of cancer. Cancer is a type of disease in which the normal cells behaves abnormally as the cell cycle becomes unstoppable and the cells keep on replicating. So cancer is a disease in which cell cycle becomes continuous and cells keep on replicating, showing abnormal growth, finally leading to death. Again, like AIDS, cancer is another disease still now the cure of which is not found. Now, what is contact inhibition? In case of normal cells, during cell division, when mass of cells come in contact with each other in a specific location, they stop dividing or replicating. So this is normal property of any individual cell. When contact inhibition, this property is lost. When this property fails, cells become cancerous. So cancer is basically a type of disease in which the cell cycle becomes unstoppable, or it's better to say the cell division becomes unstoppable and unwanted mass of cells are created or generated. So tumor, similarly, production of unwanted cell mass due to continuous cell division. After some time, in case of tumor, cell division stops. Now, types of tumor, two types, benign and malignant. Benign, that is reproduction of cells, cell division, unstoppable cell division, occurs at a specific location and may stop after some time. So basically, benign tumor means tumor which are located at a specific, the location of which is specific, means it do not spread. Okay. Now coming to malignant tumor, which leads to formation of cancer. Malignant tumor, in this case, proliferating cell mass keeps on dividing as well as attacking and damaging adjacent cell mass due to failure of inhibitors or control factors present in cell. Control factors controlling cell division. In case of malignant tumor, proliferating cell mass keeps on dividing as well as attacking and damaging, or it's better to say invading adjacent cells due to failure of control factors which control cell division. Now, causes of cancer: the physical, chemical or biological factors which are responsible for conversion of a normal cell to abnormally growing cancerous or tumorous cells are called carcinogens. So the factors or agents are called carcinogens. Now, types of carcinogens: basically of two types, that is viral Now there are also genes which are responsible, biological factors. Among that, the genes which are responsible for causing cancer are called oncogenes. Now, two types: viral oncogenes, genes which are introduced from virus into the human beings due to viral infection, which results in abnormal cell cycle, that is abnormal cell division. Those genes are called viral oncogenes. And next one is cellular or proto-oncogenes. Cellular or proto-oncogenes. So the normal cells, due to some reason, due to these genes, the normal cells get converted to cancerous or cells causing tumors. So these are called cellular or proto-oncogenes. As you can see, the chemical carcinogens present in tobacco smoke have been identified as major cause of cancer, lung cancer. As you can say that chemical agents are ionizing radiation like X-rays, gamma rays, non-ionizing radiation like UV rays cause DNA damage, leading to neoplastic transformation. Now, cancer-causing viruses called oncogenic viruses have genes called viral oncogenes. Now, normal cells, in case of normal cells, cellular oncogenes or proto-oncogenes, when activated under certain conditions, could lead to oncogenic transformation of the cells. So, chemical agents causing cancer are X-rays, UV rays, and gamma rays. Both X-ray and gamma ray are ionizing radiation and UV rays are non-ionizing radiation. Now, cancer detection and diagnosis. Several methods are used. For example, CT, that is computed tomography, X-ray, MRI. These techniques are used. Also antibodies produced against oncogenes are used to detect and diagnose cancer. Now, as you can see here, cancer detection is based on biopsy and histopathological studies of tissue, blood, bone marrow tests for increased cell counts in case of leukemia. So, in case of leukemia, that is cancer of leukocytes or lymphocytes, white blood cells, detection of cancer is done by biopsy, histopathological studies and of tissue as well as blood, bone marrow to detect the increased cell counts. And as I said, that radiography, that is X-ray, CT, MRI can also be used. Now coming to antibodies against cancer specific antigens. And now next is, if cancer can be detected in very early stage, treatment can be done. Several methods are used, including surgery, use of radiation, then example, chemotherapy, and also as you can see, radiation therapy and immunotherapy. In radiotherapy or chemotherapy, you can call, tumor cells are irradiated lethally, taking proper care of normal tissues surrounding the tumor mass. Now, chemotherapeutic drugs can also be used, but there are side effects, and but mostly cancer are treated by combination of surgery, radiotherapy and chemotherapy. Now, the patients are given substances called biological response modifiers, that is alpha interferon, which activate their immune system and help in destroying tumor. So I repeat that radiation like chemotherapy and immunotherapy are used in combination to treat most types of cancer. Also, biological response modifier, example alpha interferons, they activate immune system and help in destroying the tumor. Few more informations I want to share. Like cancer, there is no specific definition. And the first I told what is cancer, just to make it understand. But specific definition is not applicable for all types of cancer. And the name of cancer also varies according to the location. Like lung cancer, leukemia is cancer, as I told, cancer of lymphocytes or white blood cells. Now treatment also vary according to the type of cancer, but mostly surgery, immunoradiation, and radiation therapy, immunotherapy and radiation therapy are used in combination. Except than that, biological agent can be used, that is alpha interferon, which increase the immune response of a person, which is also called biological response modifier.